All right, welcome everybody. We're gonna go ahead and get started on our corn lab discussion. So you're gonna follow along with this ed puzzle, answer some questions in this to help you understand the main events of the corn lab that we did in class. First, we're going to look at the parents of the original corn that we looked at. So we've got a pure breeding purple and smooth parent, and that is going to be this RR big S, big S, big S, big R, crossed with little r, little s, which is a pure breeding non-purple and wrinkled plant. Looking at our alleles, capital R is purple, that's going to be dominant. Lowercase r is going to be cream or yellow, that's going to be our recessive allele. Capital S is starchy, that's going to be for smooth kernels, and lowercase s is our recessive allele, that is going to be our wrinkled or our sweet alleles. In class, you guys were supposed to go ahead and do some practice crosses. So the very first generation we looked at was crossing our first generation, our F1 offspring. So capital R, lowercase r, capital S, lowercase s. These are going to be a heterozygous, because two different alleles. Okay, so heterozygous, purple and heterozygous smooth generation plant. So first we needed to segregate our alleles, and this is something that we did in class together, and we found out that we have big R, big S from this segregation because the big R goes with the big S. The big R can also go with the little s, so we have that segregation here. Little r can go with big S, so we see that down here, and little r can go with little s, which we see down here. So in class, we segregated those alleles, and then we did our first two-factor Punnett square, otherwise known as a dihybrid Punnett square. So we did this in class together, and we checked this first one as a group in class. So yours should look something similar to mine. Next, we figured out our probability or our probability or statistical amount of each phenotype. So the first phenotype we looked at was purple and smooth. We had 9 out of 16 being purple and smooth. Our next phenotype was purple and wrinkled. Only 3 out of 16 from our two-factor cross were purple and wrinkled. 3 out of 16 on our two-factor cross were non-purple and smooth, and 1 out of 16 were non-purple and wrinkled. The next thing that you did in class was you did a factor cross, or you did the segregation for our cross B. So I'm going to go ahead and switch over my camera, and we are going to look at how we would do this together for those of you who still need a little extra help. So cross B was something that you had to do in class as well. And I wanted to go ahead and kind of show everyone how I'd go about this. So our first parent, parent A, we'll say is big R, big S, little s, and parent B is little r, little s, little s. So first, I like to FOIL these. There's a lot of different ways in doing this, but I like to FOIL them. Big R can go with big S, so I've got big R, big S in this square. Big R can also go with little s, so I've got big R, little s. Big R is now taken care of. I still have little r to look at. So little r can go with big S, and little r can also go with little s. On my other side, I've got R, S, S. I've R with little s. I've R with little s again. Looking at this little r, I've got little r with little s, and little r with little s again. So this is how our segregation should look. We should have had these values. So you're going to want to check your cross B and make sure you have these values. Now you might have filled in your parent A and parent B a little different from mine. I'm going to use the one on the left as parent A and this is parent B. And we're just going to be filling in these boxes. So my very first box for parent A is going to be capital R, capital S. My next box is going to be this one, capital R, lowercase s. My next one is going to be lowercase r, capital S. And then I'm also going to have lowercase r, lowercase s. That's parent A. Parent A is filled in. Next, I need to fill in parent B. As you notice, they're all the same. They're all lowercase r. They're all lowercase s. So I'm going to get all of these boxes filled in. All right. Next, I'm going to actually do my dihybrid cross. So we like to write our capital letters first, and we keep our similar letters together. So R is going to be next to R, S is going to be next to S. So looking at this first one, I've got a capital R and a lowercase r, a big S and a little s. Same thing here, big R, lowercase r, This one's little s, little s. Looking at this one, I've got little r, big S, little s. Right here, I'm going to have little r, little s, little s. Moving down to my next column, I've got big R, big S, little s. Big R, little s, little s. Little r, big S, little s. And then little r, little s, little s. So this is just an example of how we would fill this in. I'm going to switch over to my other view so that you can see what this completed two-factor Punnett square is going to look like. All right, now that I've completed the Punnett square, you're going to notice from this second cross that you're going to have almost everything the same up and down or side to side, depending on how you set yours up. So all of these were the same, all of these were the same, these were all the same, and these were all the same. This doesn't always happen, but this just happened to work out very nicely for this Punnett square. Now that we've figured out our two-factor Punnett square, the next thing that we're going to do is we're going to figure out the phenotype ratios. I like to keep a tally, so you can do tally marks, or you can do a cross-out method. Either of them works. I'll show you guys with the cross-out method how I like to do that, and you can use whatever method works for you. So our different phenotype options, and I'm going to turn my page over just so you can see our options. We've got purple and smooth, purple and wrinkled, non-purple and smooth, and non-purple and wrinkled. First, I'm going to look for how many purple smooth that I have. So in order to be purple and smooth, I need to have a capital R and I need to have a capital S. So we're going to look for any that contain both a capital R and a capital S. So here I've got a capital R and a capital S. So I'm just going to put a little mark to indicate that. Nothing here, not here, and not here. I've got a capital R and capital S here, not here, not here, and not here. I've got one here and here. So as you can see, my capital R and capital S's are all in this column. So that is 4 for purple and smooth. Flipping my sheet over, I'm going to go ahead and write out of 16. My probability is 4 out of 16. Next, I'm going to look at purple and wrinkled. So purple and wrinkled is going to have a capital R and it's going to have 2 lowercase s's. So I know that the dots are taken care of, so I don't need to look at these again. Now I'm just going to look at what I have still available. And I like to use different symbols in the boxes to help me know that that's how I counted different things. So now I'm going to do a line. So we're looking for capital R and 2 lowercase s's. As you can see, these 4 right here all are going to have a capital R and only lowercase s's. So I'm going to just put a dash in these, and that dash is going to help me designate that I've already accounted for those, and those are all similar. So 4 out of 16 are going to be purple and smooth. Next, we're looking for non-purple and smooth. This is going to be lowercase r's, but capital S's, or capital S. Looking for that, I have my lowercase r's here and a capital S in this column. I'm going to put a smiley face in all of these to know that I counted these 4. So these are my non-purple and smooth alleles, or my non-purple and smooth phenotypes in this column. So I've got 4 out of 16 being non-purple and smooth. My last one I'm going to look at is non-purple and wrinkled. So that is going to be lowercase r's and lowercase s's. Looking at my sheet, I've got my lowercase r's and my lowercase s's in this column, 4 of them. So I'm going to account for 4 out of 16 on my chart. All right, at this point of the lab, you should have had you guys are going to have different data for your counts and your count number and your phenotype ratios, because each and every group had a different kernel of corn. So I'm going to show you guys a little bit of sample student data. Your data is going to look completely different from this, but this is just an example of how you're going to do some of these calculations for this page. So you can follow along with what I did, but just use the numbers that you and your group actually got from counting your kernels. So I had a very tiny piece of corn when I did my corn counting and phenotype ratios, very little piece of corn. And my purple and smooth ended up being 56. I had the most purple and smooth. My purple and wrinkled ended up being 19. My non-purple and smooth ended up being 17, and my non-purple wrinkled ended up being 6. And that gave me a total of 98 kernels. So the next thing that we're going to do, we're going to fill in the phenotype ratio. And the way that you do this is it's our count over our total kernels counted. So my first one is going to be 56 over 98. My next one is going to be 19 out of 98. My third one down, 17 out of 98. My last one down is 6 out of 98. So go ahead, pause the video, take a minute, and make sure that you got your phenotype ratios written down with your numbers. So your numbers here and your total kernel counted down below. All right. The next thing that we're going to do is we're actually going to figure out exactly what this ratio is in terms of a percentage, because I think that this makes this the easiest for us. So we're going to figure out percentages of these counted kernels, and we're going to go ahead and write that percentage next to our phenotype ratio. And it's gonna be very helpful for us as we move forward with some of our post-lab questions. So my first one, I've got 56 divided by 98. I'm gonna take that number and I'm gonna multiply it times 100, and that's gonna give me roughly 57%. So I'm just gonna give roughly my whole number. It might not perfectly add up to 100, but we'll say 57%. Okay. 19 divided by 98 times 100 is giving me roughly 19%. And you're going to do whatever your numbers are to figure out your percentages. And I'm going to fill in the rest of mine and pause the video. So as you can see here from my sample data, I had roughly 57% of my corn was purple and smooth. This number's not right. I need to look at this number again. 19% is going to be purple and wrinkled, 17% is going to be non-purple and smooth, and 6% was non-purple and wrinkled. What I'm going to have you guys do now is I want you to do the same thing, figure out the percents for cross B, so figure out these percents, and I want you to determine the percents for your cross A as well, so cross A from over here. So I want you guys to take a minute, figure out those percentages and write them next to your cross A phenotype data tables so that you have those to reflect back on for your post-lab questions.